Get started. So thank you very much for joining us again for the SASSB's webinars. And we are very happy that today we are joined by Susan Perkins, who will be telling us about protists, and especially one of the ones that we're quite interested in terms of disease in South Africa is also malaria, but she's not only looking at one host, but so thank you all for joining. We're very happy that you could all make it. Susan, thank you for making the time and the effort to speak to us today. I will ask Kelsey now to introduce to us.
Thanks, Dennis. Thanks everybody for coming, and thanks Susan for being here. We really appreciate it. So Susan got her PhD from the University of Vermont. I met Susan when she was an associate curator of invertebrate zoology at the American Museum of Natural History in New York. And I learned a lot about systematics from Susan in terms of homology and homoplasy through discussion groups. And now Susan is a dean of science at the City College of New York, which is part of the CUNY system. And her lab group still researches the evolution, diversity, and systematics of Hemerosporidians. Sorry. So thanks, Susan.
Thank you. So thank you everybody for joining today. Thanks everybody for, and I should say apologies if you were here the first time when I missed it. To quote Barbie, time zones are hard. I really felt awful, and I'm so grateful to have another chance to share my work with you. So I'm gonna just launch right into it. I'm gonna talk about, it's really now 3 decades worth of research that I've been doing, looking at systematics as well as biogeography, and lately some genomics and transcriptomics, as you'll see, of these really interesting and super diverse parasites. So as was mentioned, you know, malaria as a human disease is still a major, I like this alliteration, major matter of morbidity and mortality. There have been some big inroads between prophylactics and prophylactic devices, things like bed nets, as well as some tentative vaccines where the cases have fallen. But of course, everyone's super worried about climate change and, you know, how that will shift the parasites, the vectors, bringing them into places that may not be particularly ready. So I've done essentially almost no work on human malaria parasites. Of course, they're super important, but they're not as interesting in some ways. And that is because the diversity across this group of parasites and the taxonomy, I'll just say from the get-go, is a complete mess, as you'll see. But the diversity is really impressive. So this slide is a little bit old. I know this is probably up to maybe 250 or more, but just within the genus Plasmodium, there were over 200 described species. And we have the 5 or so species that commonly infect humans, but we have a lot of other mammal parasites. So including a diversity of parasites in non-human primates, rodent malaria, bat malaria. You'll hear a little bit about those shortly, and then ungulate malarias as well. There were a few reports of malaria Plasmodium, I should say, in insectivorous mammals as well as pangolins, although I use this example a lot when we talk about conservation of hosts and parasites. With the critical conservation status of pangolins, I really do wonder if their parasites are even still around. Get back to my slide. We have a huge diversity of Plasmodium infecting birds, and I always choose to use this picture of a Hawaiian amakihi because it's a classic example of an invasive Plasmodium, got into a novel landscape with novel hosts and contributed to the extinction of dozens of native Hawaiian birds. And then in terms of Plasmodium, we see the largest diversity in squamate reptiles, primarily in lizards, but a few in snakes as well. And they have been found all over the world. This slide came from my PhD advisor, Joe Schall, who was at the University of Vermont. And again, it's a little bit old. We've had a bit more discovery and description of lizard parasites, but it really just highlight that we have lizard malaria parasites all over the globe. I'm sure you're honing in on the 2 species from South Africa. A few more species I know have been found there. Nico Smit and some other researchers who work in this realm have been busy sampling those lizards as well. So that's Plasmodium. When we talk about the whole family, Hemosporidae, there really is even, much larger diversity. And again, this is just described species. And towards the end, I put in a new slide that I think will drive home the point that we have a lot of work to do and there's a lot of undescribed diversity. But I wanted to just point out, so, the taxonomy has been in flux, but a genus that was called Hemoproteus, for characteristic morphological reasons, was found in birds as well as some reptiles and even amphibians. We have Plasmodium diversity, which we talked about. A lot of species of this genus Leucocytozoan, which is exclusively found in birds. And then a lot of these, what I sometimes call minor genera, oops, sorry, I took out the other slide, which are, in some cases, a monotypic genus that's been observed once, including this really unusual parasite that has been described from amphibious fish, which we colloquially call snakeheads. But a lot of diversity in mammals, including, or I should say, especially in bats. All of these parasites have a similar life cycle. There are important differences, but in essence, they alternate between a vertebrate host, which is actually the intermediate host, and then the insect, the blood-feeding insect, which is the definitive host, of course, because that's where sexual reproduction takes place. So they all enter, I'll start with the vertebrate, because of course we're vertebrate-centric. They enter into the bloodstream of the vertebrate host. If they are Plasmodium parasites, they will undergo asexual replication in blood cells, bursting out of them, producing the characteristic fevers that we associate with the disease malaria in humans, and then eventually produce gametocytes, which are then transmitted through the bite of another blood-feeding vector. Again, they complete that sexual reproduction in the insect vector. And if you just hone in on the middle of the slide, you can see we're talking about, again, a diversity of vertebrate hosts, but also a diversity of insect hosts. So it's not just mosquitoes that transmit Hemosporidian or malaria parasites broadly defined, but other things like black flies, midges, and hippoboscid flies. So there's not a lot of morphology, and I'm not going to spend much time in today's talk illustrating that because it really doesn't take us too very far. But because of this lack of morphology, pre-DNA sequencing, this was really the only tree I've ever been able to find of malaria parasites. And if you look at the top, what you notice is, I almost want to call it a Lamarckian type evolution here, where the 3 branches of this tree correspond to the vertebrate host. We have reptiles, birds, and then mammals. And some of the genera that I mentioned briefly, you see along these various branches of the tree, they were recognized as distinct because of their life cycles and their use of insects and other hosts, but it was really unclear how the relationships of these things came about. So yes, Plasmodium was the penultimate evolution of this group. So, like I said, I've been working on this group for over 30 years now, and sometimes I have to be honest, it's been a little like beating my head against the wall because it hasn't been easy. Time zones are hard, but malaria parasite systematics are even harder. And so DNA sequencing obviously was revolutionary in the sense of us being able to conduct rigorous phylogenetic analyses. But even in this realm, the malaria parasites were troubling. So I'm going to go through just a couple of these. Some of these are not new, but they're interesting nonetheless, and I hope you'll bear with me. So one of the first issues that we had was that the ribosomal DNA loci in malaria parasites in this entire group are paralogous. So our RNA sequencing was the workhorse for over a decade. It was an easy to amplify gene, and so a lot of people sequenced this gene. We know it created huge controversies in systematics because it was a terribly difficult molecule to align. But Nonetheless, it was applied to Plasmodium parasites, and Andy Waters and some colleagues got a PNAS paper out of this when their results showed, at least as they interpreted it, that we had given malaria to, or that we had gotten malaria from birds when we domesticated them. And this was immediately counteracted by folks in the systematics community, because if you actually, well, it was rooted with a completely different phylum, but if you optimize the tree correctly, the results said that we gave malaria to birds, not that we had achieved it from birds. And so no one believed that the huge diversity of parasites in birds was only, say, 1,000 or so years old from the domestication of fowl. The reason why this tree was problematic, in addition to just those two things I pointed out, is that these ribosomal genes are they exist on different chromosomes, and so they do not evolve concertedly. And furthermore, the different loci get expressed at different points in the life cycle, and so they're under separate evolutionary trajectories. Okay, so ribosomal DNA was not useful. The next set of genes that came around in molecular systematics were mitochondrial genes, and here again, hemosporidians were challenging. And that is because the mitochondrial genome of these parasites is greatly reduced. So these parasites have the smallest mitochondrial genomes of any organisms that have still retained a mitochondrial genome. So they only have 3 protein coding genes, cytochrome B, CoA1 and CoA3, and some fragmented RNA genes. And then furthermore, to make it even weirder, the 6,000 base pair locus is repeated tandemly in the mitochondrion over 100 times. But nonetheless, one of the first things I did as a PhD student was develop some primers and start sequencing cytochrome B from parasites. And so at the end of my time in Vermont, Joe and I had produced what was then the largest phylogeny of hemosporidians ever generated with molecular systematic data. So this is the overall phylogeny. I'm gonna zoom into each of the 2 major clades here and just point out some of the conclusions to keep in mind, because you're gonna see some themes as I continue through the presentation. So again, counteracting what Andy Waters and colleagues had said, Plasmodium falciparum was not falling out with bird parasites. Its closest relative was Plasmodium reichenowi, which was sampled out of chimpanzees, so not likely a recent horizontal transfer from birds. The other thing that was interesting, though, was that Hepatocystis, this unique genus for a lot of morphological and life history reasons, was falling within the mammalian Plasmodium clade, so rendering that clade paraphyletic. If we look at the other major clade, 2 major things I would say jump out at me. One is that, sorry, my phone will ring, hopefully they'll answer. One is that we saw several switches from birds to lizard hosts over the evolutionary history of this group. And then secondly, we had a very polyphyletic Hemoproteus genus. So the type species, which is the taxon at the bottom, Hemoproteus columbae, was not part of the clade of the other avian Hemoproteus parasites. So after I left the lab, Ellen Martinson, who you'll hear twice in this talk, Ellen came in and developed some other loci. So we had the cytochrome B locus from the mitochondrial genome. We also had developed primers to CoA1. I had generated some primers for a gene on the plastid genome. So hemosporidians have this remnant apicoplast organelle that predates the divergence of ciliates and dinoflagellates and apicomplexan. And then we had a tiny little fragment of a nuclear gene, ASL. So Ellen took my data, added a ton more bird data because she was sampling birds, and produced, again, the largest phylogenetic tree that had been done. And again, I just want to point out 2 things you've already heard. No evidence that falciparum was closely related to bird malaria, and even more evidence that Hemoproteus was really these 2 different genera. And so Ellen started resurrecting a genus, Parahemoproteus, that had been proposed in years past, and partly because that clade of parasites were vectored by ceratogonid midges, as opposed to the hippobossid vectors of sort of true Hemoproteus. And one of the things that was really beginning to emerge is that the vectors were the major drivers of diversification. I think this makes both evolutionary and ecological sense. Remember that the vectors are the definitive hosts. And so they're very important. Probably the original host, and us vertebrates became just replication vessels. But also ecologically, you know, they are taking a blood meal. It's an extreme physical and genetic bottleneck that is happening, sampling just a tiny group of parasites. And then if they continue to feed on hosts, they pinch that lineage down to that tiny bottleneck event so that it re-radiates out into a new group. Okay, so one of the other issues, I'm going to skip some of these things, was that when Ellen did her phylogeny, when I did mine, you know, I got to do some really cool field work, but we were limited because out of the 200 or so species that had been described, most of what was in possession in collections were just blood smears that were decades old, dried out, stained. And so it wasn't really possible to extract good quality DNA from them. And if you're thinking, what about Illumina? That didn't exist yet. This was way pre-next-gen sequencing. And so I wanted to just share a few stories from my travels and travails, which has been really fun and a highlight of my career, getting to generate some of these other samples. And I would say more importantly, getting to collaborate with some awesome folks. So here's Ellen again. That little girl is now in 5th grade, but Ellen did her postdoc at the National Zoo, and she was sampling mosquitoes, looking for evidence that they were perhaps transmitting avian malaria to the collection birds, which may be naive hosts to these things. And so she had a sequence as she sequenced blood meal that just did not fit in her phylogeny. And so she sent it to me and asked me to put it into my larger sort of backbone phylogeny. So here it is in my tree. And I know a lot of these names don't mean a lot, but there's Ellen, there's her sequence she sent me. Everything else that you see in that clade were sequences that we had pulled out of bats. And so at first, you know, we thought, well, maybe this is a North American bat malaria. But then something else happened right there at the zoo, and that is they were culling white-tailed deer because they're kind of nuisance animals in the Washington, D.C. area. And they started looking at some blood smears from these deer, and lo and behold, they observed this parasite. Now, that is a horrible photo. It's a pixelated photo. It's from a cell phone held up to a microscope. But I think if you squint a little bit, you can see that it very much resembles this species of Plasmodium that PCC Garnham, who we think of as the godfather of malaria diversity, had described from white-tailed deer here in the US. And so Ellen got a bunch of other samples. We sent her some from New York State. And it turns out that this parasite, Plasmodium oedecoilii, was here, as we termed in our paper, hidden in plain sight. Just no one had really looked at it. It has usually low parasitemia. We ended up publishing this paper, and then 2 others piggybacked off of that. So Tom Templeton, who is here in New York, at the time had some samples from African goats, as well as water buffalo in Southeast Asia, reporting, again, newly discovered or rediscovered lineages of ungulate malaria. And then Larsen Bundanga and some of his colleagues working in Gabon, sampling wild ungulates from antelope and others, again, found similar parasites. And I should say, I don't have a tree for this, but they are all in the same clade. So they're part of what I would say would be the same genus. So here's a graphic representation of that. Just really interestingly, at the same time, this rediscovery, and I say this because you'll notice there are Latin names for all of these parasites. They had been sampled back in this flurry of sampling of animals for malaria parasites, and then no one had really seen or really looked for them since. But now, of course, this opens up interesting doors for genetics and genomics as well as systematics. So there's a fun story. I don't have time to tell you the whole backstory, but just completely serendipitously, I got to meet Julian Schaar, who is a student in Germany, in Berlin at Max Planck. She was collecting some bat parasites, and we started this amazing collaboration sequencing DNA from these parasites and got 2 awesome papers out of that. We got the cover of PNAS because it was October, if you can squint and see. I think they wanted a bat for Halloween on the cover. And like I was saying, we just found a huge diversity of parasites in these West African bats. So Guinea, Cote d'Ivoire, I'm trying to remember a couple other places. And these parasites span 4 genera. So Plasmodium parasites, as well as Hepatocystis, which you saw back in the Shaw et al. Paper, which was falling within Plasmodium. And then the 2 in the middle, Polychromophilus parasites, and then Nycteria parasites. And some of these bats were very commonly infected. So 94% of a very large sample size of these Microtropis bats were infected with Hepatocystis. When we plot this across a phylogenetic topology, these are not clustering by vertebrate host by any means. So they are falling out with other parasites. But one of the things that was super, super interesting to us was that the Plasmodium parasites in these bats were intermingled with the Plasmodium parasites from African rats. And why is that important? Well, those rodent malarias are the model system, or a massive model system. Sorry, I need to jump ahead. And so the native habitat of rodent malaria these days, is white mice sitting in a cage. But these African bats are intermingling with African rodents who are arboreal. And so again, to pull in a little bit of ecology, this made perfect sense to us. Vectors coming along looking for a blood meal, feed on a rodent, and then maybe that next blood meal, they feed on a bat. And so now we've got diversification, and it looks like this may have happened multiple times. And so this is super exciting because, again, rodent malaria is a model system, but on the other side of the coin, bats are often characterized as having these super immune systems. And we say that because they can withstand these highly pathogenic viruses. And so there's a new interest in looking at these parasites from an immunological and genomic point of view. And then more recently, I've been doing some field work myself and my grad student, Rachel, on the island of Sulawesi in Indonesia. And of course, Sulawesi specifically, in Indonesia more generally, we know as just this crucible of biodiversity. That island where you see the pin dropped here, that was originally a proto-island with 5 different subgroups. Tectonic movements smashed them together, but you've had this amazing radiation of the vertebrates. And so with some funding from NSF and working with scientists from Australia, UC California, and Berkeley, as well as our Indonesian collaborators, we've done multiple trips to Sulawesi, primarily to mountaintop locations to sample. And this was some crazy field work. I love this photo. So I'm trying to wear my wellies and hike up this mountain. Those wellies were like 3 sizes too big, so not at all stable on my feet. I'm trying to balance along this culvert, and here's one of the village women helping me out, carrying this bag on her head, barefoot. She saved my life a couple times, pulling me up as I fell down the mountain. It was an amazing experience. I don't know if you can see, but the Indonesians just treat Americans like we're paparazzi, and that girl on the second one has a New York City shirt. So if you say anything about New York, even though you don't speak the same language, they think you're a superstar. So it was really fun, exciting field work, but it was also just an eye-opener, right? Sulawesi has been drastically understudied, particularly with parasitology. And my student, Rachel, who did several additional expeditions and sampled birds, her dissertation revealed 160 new lineages. So probably 160 new species of avian malaria parasites. And I just put up this principal components phylogenetic discordance analysis just to kind of highlight that, there's not a lot of pattern. The colors represent the sampling locations. The shapes represent the 3 different common genera of avian parasites. There's just a ton of diversity here. So we have a lot of work to do doing some species descriptions for sure. Okay, that was all sort of the taxon sampling side of getting a better tree. I now want to take just a few minutes and talk about how we've been adding additional genes as well. And not those kind of genes, of course, but I can't resist a pun. So another former grad student, Spencer Galen, along with a postdoc, took this set of 21 nuclear genes that had been generated by Giannis just through brute force bioinformatics. We developed these primers and amplified all of these genes from as many taxa as we could get our hands on. And this was an initial phylogeny. And I wanted to throw this in here because this is a systematics group. So when we first analyzed this with unpartitioned methods in maximum likelihood, we got a very strange phylogeny where the avian parasites, which are colored in blue, were looking like they were the more recent ones. And we had never really observed that. It looked like some of these major clades of bird parasites had diverged from these mammal parasites. And so when we applied better partition analysis, taking into account base composition biases, as well as codon position biases, this is what the tree looks like. And if you've been paying attention at home, this is pretty much the phylogeny that we've been observing overall, right? I'm going to show you just a different one that's a little easier on the eyes. But before I do that, sorry, this is why that partition analysis was so important. And that is because even though in general hemosporidians have a very high AT content, it's not uniform across those lineages. So Plasmodium vivax and its relatives are not super AT biased. However, Hemocystidium, a parasite that we find in geckos and in turtles, has the highest AT bias practically that we've ever seen in an organism. And so if that's not accounted for, it can obviously really throw off your topology. So here's the prettier tree as promised. And as I alluded to at the top of the talk, the taxonomy of this group is an absolute mess, right? And it's a terrifying thing as somebody who does not work so much on human malaria, because here is a clade that contains pretty much all of the Plasmodium parasites, but it's super paraphyletic, right? We've got a bunch of other genera in there. So we could, all right, move that clade up and allow Polychromophilus and our ungulate malaria parasites to be a different group, but we still have that problem. So what we really are faced with is that Plasmodium is a paraphyletic genus. We've got a clade that infects sauropsids, birds and lizards. We've got the Vinkia, the rodent and bat Plasmodium parasites. We've got the Vivax clade, which is in macaques, and I should say is the type species is in that clade. And then Plasmodium falciparum, and the other more recently discovered 8 Plasmodium species are in another clade, which we have subgenus names for these, but let's face it, no one's going to use that. No one's going to call Plasmodium falciparum Lavarania. So it's not been something I wanted to totally stick my neck out for. Some other interesting things, of course, ungulate malaria probably should not be called Plasmodium. We've got Plasmodium macroaceae, which was a parasite from Australia in lizards. That's not Plasmodium. That's almost certainly Hemocystidium. And then we had this really cool parasite from turkey vultures that was called Hemoproteus based on morphology, but it is its own completely unique thing. Okay. So now I want to just wrap up with genomics, right? So getting a ton more genes in order to do really robust. But even in this, lizard malaria and malaria in general has been really challenging in genomics. So the erythrocytes of lizards and birds are, of course, nucleated. And so it's not easy to separate out host and parasite DNA. And on top of that, the parasitemias that we see are typically very low. So we're already battling an odds ratio. In terms of that, the genome is so much larger in the host. The lizards and birds have small bodies. So even if we could brute force, just get everything we could, there's still not a lot of parasite DNA in there. And then if we wanted to use the other side of the life cycle equation and take DNA from the vectors, we don't even know what they are for a lot of these taxa. And so it's a proverbial, needle in a haystack. So instead of beating our head against the wall, trying to get genomics, one of the things that at least temporarily has been very beneficial is instead focusing on transcriptomics or sequencing the RNA. And that has worked partly because if we're using blood samples, the parasites are transcriptionally very active. And sometimes I would say even more transcriptionally active than what's going on in the blood cell itself. And so I'm going to just cut to the chase here and say we have applied transcriptomics to our samples and then pulled a bunch of transcriptomic data from databases, GenBank and other databases. And we produce now, doesn't even pale in comparison to my first phylogeny from grad school. So we have what amounts to over 1,500 genes, 300,000 amino acids. And yet again, if you're paying attention at home, it's really the same phylogeny, even at the same phylogeny that I generated as a grad student, almost 30 years ago. So bird parasites, leucocytozoan, Hemoproteus, Parahemoproteus, probably the original diversification into vertebrates of this group. Then we see them moving into lizards, radiating into what we now would say is Plasmodium. And I've done some tentative phylogenetic dating, and conveniently or comfortingly, it does happen around the KT boundary. We see a transition into mammals with this weird hepatocystis parasite coming out of that clade. Again, I'm just going to spend a couple minutes talking about where my lab is right now. Probably my last grad student, but she's been taking advantage of a really cool system in an amazing place. And that is this little tiny island known as Saba, which is part of the Lesser Antilles, so in the Caribbean, in between North and South America. And Saba is a really beautiful place. We call it the unspoiled queen. There are no beaches there, so it's escaped the onslaught of resorts and tourists, but a lot of divers love it there. The coral reefs are amazing, but I love it there because this is my lab. This is the dining room table at my friend Sherry and Larry's cottage where I've done my work for the last few years. So there it is, and it's home to the most adorable lizards, Anolis sabanus. But more important scientifically, Anolis sabanus hosts 3 different species of Plasmodium. Plasmodium floridense, Plasmodium azarophyllum, and Plasmodium leucocytica. These 2 we think are sister species. And within that same host, we've got this diversity. And so Giannis, who you met in the multi-gene analysis, part of his postdoc was going down to Saba, collecting some lizards, and then taking some blood samples that we could extract and sequence RNA from. We didn't have a centrifuge, so Giannis became a human centrifuge. That worked pretty well. All we were doing is getting the blood down into the tube. But with that RNA sequence data, we're starting to ask some, I think, really biologically interesting questions. So do the hosts themselves express different genes when they get infected by different either species of Plasmodium or other genera? Do they express those genes differently in mixed infections? And then on the parasite side of this, what surface proteins do the parasites express? Because that's how they decide to enter into various cells. And then one of the things that we think is really interesting now is what metabolic genes are they expressing in terms of hemoglobin degradation? And why is the Saba system interesting with respect to this last question? Well, here, let me show you what's happening. The parasites pull in hemoglobin into their digestive vacuole. They strip off the amino acids, and they're left with heme, which is toxic. It's a free radical ion, so they crystallize it into hemozoin. And so here's an electron micrograph that's been artificially colored of human malaria. There's hemozoin crystallized. We care a lot about this process because virtually all the drugs, I'm just racking my brain, that we have to combat malaria target this metabolic process. Because if hemozoin formation can be stopped, the parasites essentially poison themselves. And it's something that obviously we are not doing ourselves. So that metabolic process is extremely complex. It involves 13 different genes across, various aspects of importing it, containing the heme, and then detoxifying it. So let's go back to SEBA and those 3 parasites. We've got Floridense, which we often say is a typical malaria parasite. It infects red blood cells and it makes hemozoin. We see hemozoin crystals in the parasite cells. Plasmodium azurophyllum is also in red blood cells, but we never see hemozoin crystals in those cells. So we don't know if it's making it and getting rid of it or it's not making it. And then Plasmodium leucocytica is a real oddball. Even though it's a plasmodium, its sister taxon is Plasmodium azurophyllum. It uses white blood cells as its host, and it doesn't make hemozoin. So we really were interested in that group, of course. So this is work done by my PhD student, Sarah Pangburn. So she has taken different clusters of the genes involved in hemoglobin degradation, and again, presented them here as a principal components plot. You can see that the expression clusters and these genes themselves do fall into very distinct groups based on the parasite taxon that those genes are found in. So they're expressing them differently. The heat map is a little bit messier, and I would say that has some oddities, or at least things I don't quite understand yet. So we see upregulation, the dark red colors, and the green on the top represents Plasmodium floridense. That's expected because these parasites we know are making hemozoin. But we're also seeing some upregulation of particularly this protein vasolysin in Plasmodium leucocytica, the white blood cell parasite. So that's a little bit strange. But one of the questions that Sarah is attacking now is looking for evidence of relaxed selection. So our phylogenetic analyses suggest that azurophyllum and leucocytica, these 2 parasites that don't produce hemozoin, are more recently diverged off hemozoin producing parasites. And so maybe they are in the process of pseudogenizing or even losing these genes. So they still have them, they may still express them, but are they still under the same forces of selection? And so far, Sarah's preliminary work has said no. So when she compares azurophyllum to floridense or leucocytica to floridense, we do see some evidence of relaxed selection in these hemoglobin degradation genes, again, suggesting that they may be in the process of losing these since it doesn't look like they're using them. So I'm just going to wrap up with a few conclusions. I hope that you learned a few things through this talk. I hope you enjoyed hearing about this. So contrary to a very popular and often highly cited paper, malaria is originally a parasite of birds that probably just invaded mammals once, and Plasmodium falciparum did not recently jump into humans from chickens. We don't understand some of the convergences between falciparum and avian malaria, but that doesn't seem to be a relic of evolutionary history. The taxonomy of malaria parasites still needs a ton of work. We've got a lot of genera, some of which are poly and periphyletic. We know we've got tons of just basic biodiversity species description work that needs to happen, not to mention a ton of sampling. We know that there are a lot more parasites out there to be discovered. So the diversity of the type of host they use, and that's vertebrates as well as the insects, the type of host cells they use, different life history traits, including the tissues that they infect and the life cycle timing, as well as the pathogenicity that we see, offers an amazing potential for really true comparative genomic studies in this group. Throughout the last, I would say 15, 20 years, we've hit a lot of brick walls with genomics, but transcriptomics is offering a lot of promise. And I should say there have been some breakthroughs more recently using some of the newer DNA sequencing techniques as well as separation techniques where there have been some genomes generated for some of these parasites. I want to just acknowledge a lot of folks and agencies that helped with samples, with logistics, with helping out with bouncing ideas off. As Kelsey said, I spent about 15 years at the American Museum of Natural History, benefited a lot from funding that I received there. NSF has funded our work in Sulawesi as well as other work. The NIH funded some work that I did with Diana Outlaw, who's cited here, and then some work with the Australian government getting to sample there. And I realize my conclusion slide is a little old. It still has the old Twitter logo. I still venture onto, I guess, the site normally known as X now, but I'm very happy to stop sharing what I have so far and entertain any questions you might have. Thanks.
Thanks, Susan. Does anybody have any questions before I start asking?
There's no hands at the moment, so I think you can go ahead. Kenneth,
I see
Kenneth,
Kenneth Fan. I'll give him a chance.
Yeah, hi folks, can you hear me?
Yeah.
yes.
Thanks, that was really cool. My question is one from pure ignorance. So I think those trees showed fairly obviously that there's been movement from birds into mammals, but do we happen to know where did the original Hemosporidian parasite invertebrates come from? Has there been any sort of larger scale sort of phylogenetics with regards to the host jumping at larger scales?
So based on the samples we have, the data we have, at least in terms of taxonomy, I would say it was probably in Africa and probably from a bird into a bat. I really think, and others have pointed this out in other analyses that they've done, that bats have been this really key transitionary host. And so the bat parasites are, at least again, it's only based on the samples we have. So I want to, put an asterisk on that. But the earliest diverging mammalian lineages in our trees and others who have worked on this are in bats. And again, I tried to highlight that we should be thinking of ecology here sometimes. And so, I mean, my guess, and you can tell Jonah this, Kelsey, that dinosaurs probably had Hemosporidians. We don't have great fossils. We have a couple fossils of insects from amber where you can see the oocysts, these stages on them, but we have no idea what they are. I mean, we know it's a Hemosporidian of some kind making an oocyst because they're the only ones, at least currently, that do. But what that insect was feeding on, there's no way to know. But that dating puts these things back, into the 100 million years or more. So they very likely infected, at least the dinosaur lineages that the avian, proto-avian lineages, and then maintain themselves in avian hosts. And then again, I think ecologically, it makes sense that if they were looking for a warm host in a tree, they might bump into a bat. But the whole thing is just amazing to me because bat blood is mammal blood, right? Is so very different than sauropsid blood in all kinds of ways, biochemically, cell membrane, nucleated, metabolism. So somehow this has to have happened and it worked, right? And it just kind of blows my mind. As we go through the mammal parasite phylogeny, you do see, as I tried to show, bat parasites are scattered throughout, and oftentimes they're paired with other lineages. So our results seem to indicate they were in rodents first. That's probably, it could go either way. There's probably missing bat lineages. Bat parasites are sister to the ungulate malaria lineage. So I think for two reasons, which are just broad, bats are super diverse, right? And ecologically diverse as well. There's just been a lot of opportunity for Hemosporidian parasites to infect bats. And then maybe also coupled with that, their motility has brought them into proximity with other groups of mammals. And sometimes those host switches work. So.
Thank you. How cool.
Yeah.
Thanks. I think that you almost answered the question I had, which is what about the vectors could make the parasites diversify? But I never really thought that like mammal blood and sauropsid blood would be different, because as a plant person, blood is blood, right? So
Yeah.
And so
Yeah.
so what could these things be responding to?
Well, I think there's two things. And again, one thing is maybe a bit more ecological, and that is, ever since I learned, it's really the life history on the vector side. So, let's talk just a mosquito comes along, takes a blood meal. She needs those proteins and fats to lay her eggs. If she picks up Plasmodium, it's developing within her midgut, but it's kind of ripping her up, right? So it's chewing up her midgut, and then she's got parasites in her salivary glands. And something like 1 in 1,000 mosquitoes live to take a second blood meal. So there's already this massive bottleneck happening of vectors that pick up potential parasites surviving long enough to transmit them. And so part of that difficulty, I've always suspected, and I've never done the experiments, I've talked about it, is that I wouldn't be surprised, I'll just couch it like that, if vectors have a little bit of imprinting in terms of where they found that blood meal. In other words, I wouldn't be surprised if those that do survive tend to go back to, the source, if you will. So whether that's a microhabitat or the odor of a host or whatnot. I mean, that's not ubiquitous. We know of lots of mosquitoes that, will feed on lots of different hosts, but I wouldn't be surprised if that's played a role. And so you're talking about just what could be an utter bottleneck of a single lineage that now is starting an infection. And then there's all of these places within the life cycle for diversification of genes to happen, right? So they're asexually replicating. You've got this multi-part mitochondrial genome where you can generate diversity, right? You've got the within host selection. So, I mean, I wish I knew more mathematics and statistics, because this is such a challenging problem to model, right? All these steps that are going into this. So yeah, I think it's just the fact that you're probably shrinking it down to a lineage, maybe 2, and then an opportunity for just an expansive diversification, especially if they can continue to infect those hosts and now are under new selective pressures. So.
Cool. Well, thanks, Susan. I now have a few more stories to tell my first years when I lecture them about protists.
Yeah.
Are there any other questions? If not, oh, okay, yeah.
You can go ahead.
Okay.
In the species that you're looking at that infect these wonderful lizards in your lovely little holiday home, is there a different time frame in which your data shows they started to infect these lizards? And might that have some bearing on what you're seeing?
Yeah, that's a great question. So some of the work I did as a PhD student was more specifically on the phylogeography of Plasmodium azoriphyllum and Leucosidica, right? So Plasmodium floridense, and I should say one of my former students showed this is probably a multi-lineage group. But that parasite came to the Caribbean from, it has a distribution all throughout the Caribbean basin. So we see floridense or its very close relatives in Mexico, throughout the Southeast US, and now it's infected the northern islands of the Caribbean. Azoriphyllum looks like it probably had a slightly similar phylogeographic past. And I mean, I don't know how much to totally trust these data, but Leucocytica looks like it arose within the Lesser Antilles, that it diverged off very recently. So yeah, I mean, they're different ages. They've infected other kinds of hosts. But my suspicion, honestly, is that Azorafilm and Leucocytica, even though some of my early data showed that they might have come up through the Northern Caribbean, their common ancestor is probably some of these parasites that we now can observe in South America. And they are more commonly, they sometimes have gotten lumped with leucocytozoan because they don't make hemozoan. They look somewhat similar. So I don't know if I'm totally answering your question. It looks like they're more recent, and because I have limited sampling, they look more closely related to the hemozoan producing parasites than they probably should be. And that's only because I don't have great taxon sampling from their true closest relatives. But it's been an interesting system to work on because, I mean, I choose to work on Ceiba. I've been working there a long time. It's an easy place for me to work, not just beautiful. But it's replicated. So you have other Anolis hosts throughout those islands that are also infected with the same 3 species. But they probably haven't been on those islands very long. There hasn't been a long time to diverge, mostly because those islands are not terribly old. They're 8 to 20 million years old. And then layered on top of that are massive hurricane storms, at least over recent history, which we've observed wipe out whole species of parasites on these islands. So yeah.
So the other thing that struck me was that with the parasite deriving so many amino acids as sources of nutrients for all sorts of pathways by digesting hemozoan, what's the alternate parasite that doesn't use hemozoan at all getting for obtaining all these amino acids from? I think that biochemically is very interesting.
Yeah.
Change in metabolic patterns.
What's cool is that, mammalian Plasmodium, all they have to digest is hemoglobin. That's the only protein in our red blood cells. And so part of that is historical. We know tons about this pathway because it's studied so intensively in human blood and human malaria, because it's all they've got. So all of these proteins and hemoglobin metabolism, note my air quotes, right, were studied in falciparum cultures. But my guess, again, we think these parasites originated in birds. They have nucleated cells. Hemoglobin is not the only protein in sauropsid blood cells. And so probably most of these hemoglobin digesting proteins are just proteases. They're just proteins that the parasite uses to break off amino acids because it needs those as raw nutrients to create its own proteins for growth and reproduction. And just we've been so focused on human malaria that we only see it through this hemoglobin pathway. And so I think Sarah's data and some of my other students that worked on this are a good reminder of the fact that things that we see as for a sole purpose may just be a historical artifact because of the system in which they've been studied. And so we see all of these genes. I didn't put that exact data in there, but every single one of those genes in the hemoglobin degradation pathway are still expressed in every parasite we've ever sampled. And the lizard and bird parasites, again, have access to more than just hemoglobin. And so I think it is really interesting biochemically because we don't know those details of what, are the lizard and bird parasites specifically using specific proteins. Again, mammal parasites have to use hemoglobin, lizard and birds don't, but do they have a preference? Why? What is it about them? There's tons more questions, definitely.
Thank you. Lots of food for thought.
Yes, literally. I was gonna say, and speaking of food, you all are probably getting hungry because it's getting close to tea and happy hour and
So, well, great. Thank you so much, Susan. That was really interesting. I knew you worked in the Caribbean with lizards and malaria, but I had no idea how exciting and interesting it was. So thank you for sharing that with us. And have a great rest of your day. So,
Yes,
and thanks everyone for
Thank
coming.
you for inviting me, and it's so good that we could make it happen and got the time zone.
Time zones are hard. I do agree
Really
with you.
tricky, and especially when we switch them all the time here. So yeah.
Yeah, daylight savings.
Dang it.
So cool. Thank you.
Take care, everybody. Thank you.
Yeah, from my side as well, thank you for really a great talk. As a bacteriologist, I really also enjoyed it because taxonomy is taxonomy. And ecology is ecology, and it makes life quite interesting. So thank you very much, and also to everybody that joined today. We are very happy that we had quite an audience today to come and listen because it's really great. And we will post this talk on the website, the YouTube, so you can tell your friends that it's something worthwhile to watch. Thanks a lot for your time and enjoy your day.
Bye,
Thanks.
everyone.
Bye.